So, welcome students. Yuvraj, invite others also to join.
Yes, sir.
बिल्कुल आपने स्लॉट्स प्रोवाइड कर दीजिए लेकिन आपके कौन से स्लॉट्स हैं वो टाइम टेबल कर रहे हैं बट स्लॉट हमारी बात बिल्कुल लेकिन मेरे को आप कर के दिए ना उस हिसाब से राइट से कि क्या करें आप एक काम करिए स्लॉट्स हाँ कोई बात नहीं वो टेंटेटिव रहेगा लेकिन आप स्लॉट्स प्रोवाइड कर दीजिए हाँ हाँ मैं मतलब इसको कंसिडर करके चलें अभी हम टाइम टेबल अपना भी कर रहे हैं यस take it up then I think the tools will be taken up at lower level. But we'll see that timetable आप slots भेज के एक बार देखते हैं हाँ वही वही that we need to confirm आप एक बार slots भेज देना ये तो भी मुझे मैच ये न्यू साइट है वो फाइन उनके ग्रुप में ये जरूरी कुछ slots है slot में सारा हो जाता
Uh, okay, students, welcome back. We were talking about. Am I audible well? I'm well audible to all. I'm audible to
Yes.
all.
Yes, sir.
Yes, sir.
We were talking about southern blotting in our previous class, and we also talked about the labeling, how we are going to label your probe. So in the labeling, we talked that mentioned that fragment we are going to utilize it. Bit history, we talked about who discovered it. The name of the Edwin Southern, we are naming it Southern Blot. And this thing we have done, and then we talk about how we to hybridize probing, etc. And what frequency when you're talking about, and that's also things. So restriction digestion we do in that case so that the fragments forms, and then we transfer it to the membrane, and membrane is the capillary action. You can say this is the way how is a rough design of your southern blotting, how to perform. So below the sport is there, then you place your gel, and then you put your nitrocellulose membrane or nylon membrane, whatever membrane you're going to use it for. Then you put a stack of paper, and then over it you put a weight. So these paper towels, they pull the capillary, help in the capillary action. So with the capillary action, your DNA moves to the membrane. And so this is the way how you are going to perform it. So southern blot, this is just a graphical representation how it's really happening. And then after this, the main thing got when the nitrogen move on, then you need to bake it, and after baking it, so you need to hybridize it. Hybridization on the basis of the complementarity. So that this sequence specific, shape specific, these type of things. So hybridization and the complementarity both more or less similar things. The main thing when you're talking about here is you are hybridizing with your probe, and the probe is going to be DNA, RNA, or a protein. So hybridization can be protein, DNA, and RNA. So this is about this brief what we have talked about the southern blotting, and in previous class we also talked about some specific procedure also how to perform those different things and what are different steps involved in this southern blotting. And now we have to move to the next step, that is the northern blotting. And northern blotting and southern blotting both are similar thing. The difference that we're talking about here is going to be your when we are transferring in case of southern blotting, you're transferring your DNA, but here it is the RNA. So it is very much similar to the northern southern blotting. Nothing different is there, but it was developed by James Alvin, David Kemp, and George Stock, and they name it northern just to respect to the southern. Just that's the only thing. Nothing. The name northern blotting and the southern blotting there, because the same techniques are more or less similar. Just only the difference is using mRNA, or using RNA, or using DNA. So the probe here also going to be the complementary thing. So probe here also whether maybe DNA or RNA. As in case of southern blotting, your probe may be DNA or RNA. Here maybe the probe. Only the substance that you are transferring over the blotting paper or your membrane, that is your RNA in this case, and in case of southern it is DNA. That is only the basic difference. Otherwise, technique wise both things are similar. And transfer how we are going to transfer, that may also differ here because where you are running your mRNA, that also be an issue. So as coming to things, what is northern blotting? So northern blotting is a blotting technique used molecular biology to study the gene expression, utilize detection of RNA, also known as RNA blot, because we are blotting the RNA, we call it the RNA blot. But commonly, as it is similar to southern, we are calling it as a northern. So it's not a big difference. So whatever things we are talking about here, the schematically, here also the same thing. So in the previous case, southern blotting, we are taking the cleaving DNA. So here you need not to cleave the DNA. You are directly going to have an RNA. So RNA is a smaller thing, so smaller in size. You can simply just transfer the RNA. All the setup you can use the same. So where that you are transferring DNA, here you can transfer the RNA. So this is only the main thing going to differ. So just you can see where you are seeing here in DNA, you can replace DNA with the RNA. So that's the only difference. Otherwise, rest all things are similar. So you may say that sometime what happened is when you are running RNA, in the case of RNA, we generally do not run your agrogel electrophoresis because your RNA size is a bit small. If the larger RNA you are talking about, then it's fine. But if the RNA is very small in size, then it's not DNA agrogel electrophoresis doesn't allow you the separation of your RNA. So in that case, what we are doing, we are running the page polyacrylamide gel electrophoresis. So from the polyacrylamide gel electrophoresis, transfer from the polyacrylamide gel to your membrane is a bit difficult process because the pore size in case of your acrylamide electrophoresis are very small, so the capillary action won't allow the migration of your particle through those so small pores. So in that case, we can use the electro transfer or electro blotting rather than your normal capillary action blotting things. So principally wise, blotting is method used to study gene expression to detect RNA. So main simple thing you can say here you want to detect your RNA. So whether your sample is going to have your RNA of importance or not. So RNA of importance means RNA only form when the transcription is going to occur. So otherwise RNA is not going to form. So whether in the cell the transcription is going or not, that is the main aim to study this northern blotting. In case of your southern blotting, DNA is going to be remain there whether the transcription is happening or not. But in but in case of the northern blotting, whether the transcription or if the transcription is there, then only the RNA will form. Otherwise, RNA will not form. So simply, you can say that when we are talking about this northern blotting, we are going to talk about some specific type of gene or specific type of condition, whether the gene is expressing at that time or not, and the specific to tissue also. Like if you talk about your skin tissue, your liver tissue, your eye tissue, your bone marrow tissue, your blood. So in every type of tissue, the expression of your genes are going to be different. Like simply if you talk about in case of plant, in case of leaf, it is going to be different. In case of root, it is going to be different. In case of stem, it is going to be different. Not just that, even if you compare with a different time interval. In morning, if you take a leaf, then it is going to have different expression pattern. If you talk about your
Sir, you're not audible. You're on mute.
sorry, so we are talking about that how you're going to get this RNA into your sample and how you're going to get that and what is going to be the implication, what is going to be the usage of this technique. So this technique is going to be tissue specific and sample specific and time specific. So each and every time, like if you say in the morning, your the photo respiration, your photosynthesis is going to be very low. So those genes which you need for the photosynthesis in the morning or in the night. And you also know that like in plants you might study two different type of reactions are there, day reaction and the night reaction. So during the the enzymes that you need in the light reactions are going to present whenever the light is going to be there. When the light is not there, those enzymes definitely not going to be there. So that you want to study that pattern. So northern blotting is going to help you, but southern blotting won't help you in that case because DNA is going to remain constant whatever type of cells you are having. So how to perform it? The performing, as I mentioned, is similar to your DNA. So if you want to have the agrogellular electrophoresis, you can simply perform the capillary reaction for the DNA transfer. What are the setup we are talking in case of southern blotting? The same setup is going to be here also. But if you are going to use page polyclonal gel, then you need for the electro blotting. So in the electro blotting, I will show you the instrument when I will talk about the western blotting. So this is so then you have to follow the hybridization when the label DNA probe. It's simple contain complementary sequence. So this is the how to the probing and how to you hybridize it. So that's coming to the form of detection. So detection of RNA, if interested, you can go for the chromogen also, like any rotative means depending on the type of probe you are having. So if it is a radio level probe, you can go for the radio labeling. If it is a chromogenic probes, then you can go for the color development, like some enzyme is there in that case. And if it is a fluorescent probe, then you can go for the fluorescent. This is the all way you have to want to visualize what type of probe you are going to use it. On that basis, detection is going to be there. Then application, as mentioned, the application way, its main application of this is gene expression, pattern tissue, organs and development stages, environment stress levels. All these things are different way of describing the expression. So like a different tissue are going to have different expression pattern of a gene, environment stress is there, so environment stress, then you can say that like simply if you are going out in the sun, so your heat stress, so what type of the genes expressing in you in the heat stress. Simply in case of cancer also, what type of genes which are specifically expressing in your cancer patient, like what are the tumor suppressor genes or tumor activator genes, and what are the level of tumor suppressor gene in case of cancer, how they are going to be regulated or down regulated. So all these you can study in using your northern blotting. Here, like in case of recombinant, if you are forming recombinant also, like RC recombinant DNA technology. So if you make a recombinant also, you make a transgene, but whether that transgene is showing its expression or not, that you can say by the checking the your northern blotting. Because if sometime happen that if you make a transgene, you go for a southern blotting. Southern blotting will tell you whether the transgene is formed or not formed. So southern blotting confirm that a transgene is there, but still you are not getting any protein, any expression of that gene. Then at what level that expression is being blocked. So whether the transcription is occurring or not. So if you want to check whether transcription is occurring or not, you need to go for the northern blotting. So if you found that northern blotting is showing the presence of that mRNA is there, but still no expression is there, then you can suspect that now it is there at the level of the translation, not at the level of transcription. So even at the then to want to confirm whether the gene is there or not, for that you need to go for that level for translation. So for studying that, the next technique is going to be there, that is called the Western blotting. Okay, so another application is the reverse northern blotting. What this reverse northern blotting is that this is the technique to check whether means here what you are using here using a DNA and want to check whether that mRNA is there or not. So reverse blotting method, which gene expression pattern may be analyzed comparison of isolated RNA molecules from test sample or a sample controlled cDNA library. It is a variant of northern blot in which nucleic acid immobilized on a membrane is a collection of DNA. So what simply can say, you are doing a southern blot, simply you can say, and the southern blot you are blotting with the mRNA. So mRNA is going to be expression. So whether that gene is present in your DNA or not. So by taking an RNA, like if you extract RNA from me and take my RNA to check whether the whatever the genes are present in me, whether that gene is present in other or not. So to this you call the reverse northern blotting. So you are blotting DNA, then over you are putting mRNA, and then probing using the probe, you are checking whether the mRNA is there. So this technique can go for the reverse for finding the gene of an mRNA also, whether the gene is there in your cell or not. So this is a simple northern blotting, and now come with the advantage and disadvantage of northern blotting. So advantage is simply you can check whether the expression is there or not. So and only mRNA from the cell type that you need, are there, because mRNA are the one which synthesize protein. So we can hybridize them. So you can also use the protein for the hybridization here. It has useful detection of mRNA transcript size, specifically really high. RNA splicing visible because alternative splicing transcripts can be detected. So you can find the splicing is there or not. So before the splicing of spliceosome, that type of study you can do here also. Once you bloat it, you can store it for a bit larger period also, because once you transfer to the membrane, so the chances of that mRNA is a bit more susceptible to degradation. So once you transfer, then it is uh, it is protected. Simply you can say that. But this is also disadvantage of this normal blotting, because at the start step, the risk of degradation is there during the electrophoresis itself. The quantity of expression is very much affected by even the electrophoresis or the reagents that you are using. So whenever you are using any reagent, in case of whenever you are handling RNA, all the reagent must be treated with the DEPC, and this DPC is for the that degrade all the RNAs or the nucleases that are present. Hydroxide of radioactive and formaldehyde are risk of poison. So these are the this is the disadvantage of every type of the blotting. So but here the formaldehyde you are using, that may be another thing other than the radioactivity. So these are the few risks that you need, disadvantage of your northern blotting, but the aim that you can get from the northern blotting is not possible with the southern blotting. But if you want to avoid this northern blotting, you don't want to go for the RNA, then you can go for the directly for the protein, whether protein expression is there or not. But if you found the protein expression not there, then you can go for the northern blotting to check at what level the problem is. So here are some precaution while you are performing your northern blotting, that you need to check whether the air bubble is trapped in the gel, because air bubble, as mentioned, the capillary action we are performing, and even the non capillary action is not there, we are performing the electroblotting. Even in that case, the presence of air bubble inside your between your gel and membrane create a conduction error. So electricity will not be conducted, and you may add to the loss of your sample. And so the electrophoresis tank are rinsed with the distilled water after use. So because if any previous residue is going to be there, then that lead to the degradation or the contamination of your sample. Control temperature during hybridization, because DNA is DNA can be can get, sorry, hydrogel can get melt if you go for very high temperature. Always check the incorporation of radioactive well before using the probe. Don't reuse electrophoresis buffer, because electrophoresis buffer will lead to the degradation of your RNA. And the radioactive probe also you should not use every time, because that they may also contaminate your, or they may also be the source of degradation of your mRNA. Next type of the technique that we are talking about is the Western blot. Western is another, as when you are talking about a name, North, Southern, Northern, and Western. So this is a just continuation of that the naming is, but here you are going to utilize protein, and the protein for detection use the antibodies. So that is also for the immunoblot or the Western blot. So who is the pioneer of it? So W. Neil Burnett in 1981 he discovered this technique, What you do, you discover the presence of specific protein in given protein sample. Procedure is this is also similar to what the procedure talking about the southern and the northern blot. And the only difference is that there you are talking about the DNA and RNA, but here we are having the protein or the cellular extracts that is that contain the protein. So probe is also required here also. There also radioactive probe we are talking about. Here also probe we are talking here also. Similar three types of probes can be there. That is the radioactive probe and your fluorescent level probe or a chromogenic probe. So all three types of probes as were there in case of your DNA and RNA, your western and your northern and southern. Similar type of the probes are going to be here also. But the probe as in case of DNA and RNA, we are talking about their DNA and RNA probe, but here the probes are going to be protein probe. So protein-protein interactions are going to we are going to talk about, and protein-protein interaction that are here is the antigen antibody interaction that we are talking about. I think you all are studying immuno course in this semester definitely, or not if not in this semester, definitely into the next semester we are going to study that. So simply just at a level, I want to tell you that antibody and antigen they are having specificity, and the specificity is as much that you can say that one antibody per antigen is there. So every antigen has its antibody. So no two antibody interact with the same antigen, but it's not always true. Sometimes you have a cross reactivity, means like antibody for A can cross react with antibody for B also, but that happen only in a case when there is a very much structural similarity. Otherwise, no two antibody interact with the one antigen, or neither one antigen can interact with the two antigen. And this way, one more thing is there if you like to study, can study that is a polyclonal antibody and the monoclonal antibody. So polyclonal and monoclonal antibodies are another concept which we will study. So based on this, the protein-protein interactions in the ELISA, also in the immuno course that you are going to study, that's the enzyme-linked immunosorbent assay. So it is a Western blot is also just a modification of ELISA, you can say, because in ELISA you are using antigen antibody complex. So here also we are going to perform antigen antibody complex. So there we are using enzyme is there. So here also are antibodies that we are going to use for detection going to link with the your antibody. So protein denatured with the SDS that you need to check. So how to perform it? So as I mentioned, it is similar as case of your northern and southern blotting. In that case also, you first you perform the gel electrophoresis. Here also you are performing the gel electrophoresis, and when you are saying the gel electrophoresis, here the gel is going to be a different type. In the previous case, we are talking the closed gel, but here the gel is going to be polyethylene. So the size of protein is very small as compared to your size of DNA or RNA. That you can simply say in a way that your protein is 3 times smaller than your DNA. So DNA is going to have a 3 nucleotides that represent one amino acid. So third 3 times smaller than in this way. And second thing, you can also say that check that your nitrogenous bases are big, very large in size. They are having they have sugar component, they have phosphate group, they have nitrogenous base, so that make it bulky. Where our proteins are bit very much smaller than the size of your nitrogenous or nucleotide. So proteins are very small, very small in size as compared to your nucleic acid. So that's why we cannot run in the hydrogel electrophoresis. We can separate them better in your polyacrylamide gel electrophoresis. So when similarly as you perform the hydrogel, you are performing this also. So once the electrophoretic band you got, then the next thing is a transfer or the blotting. So you mentioned there you are performing the blotting just by plate or a capillary action. Because the pore size is a bit large in case of your echo gel, but here pore size in your polyacrylamide gels are very much small. So we can't perform a transfer in just by use of capillary action. We need a specific instrument. So in left you can see here there are two different two different this is a semi-dry one and this is going to be a wet transfer. So you are going to have electrophoretic tank and in this their cassette is there. So in this cassette you put your gel, then overlay that gel with your membrane, and then here also you have to place all those things that you are doing in the case of your southern or northern blotting, like your blotting sheet. So the capillary action will also occur here, but it won't be very fast. So then you have to place in this cassette into the slot, and this slot you put into the your electrophoretic tank, and you are going to have some buffers in it, and negative and positive you have to connect it. So protein being negatively charged, it should move toward the your gel, so you can that will help to make your transfer. And here you don't need so much cross linking, because amine group are present in your protein and amino group are present in your nitro group present in your membrane. They facilitate the linking, and the charge also make them linking firm as compared to your DNA, and DNA with larger size can penetrate into the membrane. So here that can also happen. Okay, so the protein binding is based on the hydrophobic interaction as well as the charge interaction between the filter and the protein. So this is the way how these two are interacting. So here you don't need such blocking or cross linking as you're doing in case of DNA and RNA. Okay, and so then the next step is the blocking. So blocking, as we're doing in case of your northern blotting and southern blotting, here also you go for the blocking. So blocking, there we are doing it by using the DNA or RNA. So here also we do the blocking as we want to block the protein. So here blocking to be done with the protein only. So for the blocking you can use two different types of protein. One is either you can use milk. So milk, generally we are using a skimmed milk, or that milk powder, the Nestle, Nescafe, or the Amul, any milk powder that came. So that milk powder we use for the soaking or the blocking. And if you don't want to use the milk powder, you can use the BSA also, bovine serum albumin. So they block non-specific regions. So where your probe and your antibody want to go and bind. Then protein solution we bind the main function prevent the antibody from the binding to non-specific to nitrocellulose and the unrelated protein of the nitrocellulose. It increases the probability that they might immobilize antigen protein. So it facilitates blocking is to facilitate binding of antibody to the your antigen or your protein of interest only, not any other piece. Now once you transfer, once you block, then the job is probing, and probing is probe here is our antibody. And antibody, can we use the two types of antibody we use. One is the primary antibody. Primary antibody is one which binds to your antigen or your protein of interest. Okay. So, but the problem with this primary antibody is that we have this antibody specifically, and the amount of this antibody are limiting. So we can't produce this antibody in a very large amount, because for each and every protein you have to produce this antibody. So for that, what we incorporate one more antibody that is specific to your primary antibody. Okay, so the secondary antibody is not going to be specific to your antigen. It is going to be specific to your antibody, because antibody is also a protein. So what you are doing in this way? In this way, you are first binding your. So what is going to happen in this case? Just a moment. So what is happening in this case? Like, this is your protein. So now to this protein, first your antibody go, and it is antibody is going to bind here. Okay, but this is also protein. This is also protein. You can't say. So what to do? As mentioned, we have to make it a probe. So we need to label this with any chromogenic, fluorescent, or radioactive substrate. But this is very limited in amount, so we can't do that. So what we are doing, we are making one more antibody, and this antibody is going to be specific for this. Okay, so this antibody will go and bind at this location, and this antibody will bind to your protein. So this is going to be primary. So primary one is a specific one which is going to determine whether your protein is there or not. Now we are adding the secondary. So if the primary is bound to the protein, bound to your target, then secondary will come and it will bind to the primary antibody. This secondary antibody you can produce in a large amount. Okay, so this is specific to this. Like if we are producing this antibody in mouse, So then this is going to be mouse antibody. So you can raise anti-mouse, means any antibody against the mouse antibody. So this you can raise even in the goat and rabbit, horse. So from there you can get in blood in a large amount. So then if you can work with them easily. Where the mouse total volume of blood is only 5 ml. Out of 5 ml, when you are going to take out all the blood that is present in mouse, you are going to end with at the max 3 ml. And this 3 ml volume you are having very little amount of your antibody. Then but for this antibody, the horse having the blood in liters, 10 liters, 20 liters. So at least one out of 20 liters, 10 liter blood that a horse is going to have, you can take 1 liter of blood. So it is not going to sacrifice the life of your horse also. But here if you take out the 3 ml blood from the mouse, your mouse is going to die. So maintaining the life of horse, you can take out 1 liter of blood. Like if you talk about human, we have the 5 liter of blood. So when you are donating, you are donating around 500 ml of your blood. Okay. So if you, it is easy to take out. We are not going to die if you take out the final blood, but it is going to be very larger amount than the blood of the mouse. So the primary antibody we can use in the mouth, that is going to be very small amount, but the secondary antibody which we can commercially get from the companies which are going to be against this secondary. So this large amount and large amount, this you can label. Label you can radioactive label you want, you can do that. You can label with the chlorophore, you can label with the chromogenic like enzyme. So for chromogenic one, this is the HRP is the enzyme that in the horse radish peroxidase. So this enzyme is conjugated to the secondary antibody. This antibody will go and bind specifically, like if the antigen is not there. So if this primary antibody is not going to bind, then secondary will also not bind. So wherever this interaction is there, the secondary antibody will give the reaction, and it's an enzyme. It's on its chromogenic substrate like DAB, diaminobenzidine. If you put it with this and H2O2, then it will give a blue color product. Okay, so you can see that the blue bands are like if the protein band which is present on your gel, if it become blue in color, and simply you can say that there is a protein is there. Otherwise, it is going to be uncolored. Okay, so this is the thing. Now you can visualize color reaction. Signal both minor and major antigens can be visualized, optimized on the base on a single plot by varying exposure time. So if the very less amount is there, then we are going to for the detection by increasing the exposure time. So this is simply how to visualizing. Visualize I mentioned the homogenic thing is there. So there how it is going to occur. So as I mentioned, the primary and secondary antibody. So all things are present. So this is your protein. This is going to be your primary one. This is going to be secondary, and this is going to be your enzyme. This enzyme cut on your chromogenic substrate with a color, or if it is chemoluminescence, then it produces like it's all depend upon what substrate you are taking and what product. Whether the product is going to be light, then it is going to be light, and if it is going to be color, then it is going to be a color. So by this, we done with our all the type of the hybridization of the protein, northern, western, and so now before closing and before coming to you for the questions, I would like to show you some video also where you can really see how this experiment, how really we are performing it. So just wait for a moment, I will share you the video. Okay, so is video available to you? Size and relative abundance in the sample. Is video visible to you?
Yes, sir. It's visible.
Yes,
Using this buffer to equilibrate your gel prior to starting the Western blot. Next, so this is a polyexperimental you have taken out. The opening key. Line up the arrows on the lever with the 4 arrows on the cassette to open the cassette. So this is what type of gel you are having. So this will first you soak into your pre-transfer buffer. Now these are soaking. These are the pads that you need for transfer. They also need to soak into your pre-transfer buffer. This is your cassette. How to set it? So this is a wet transfer you are making. I'll show you the image.
Place the gel holder cassette in the container with the black side down.
So here also the color thing is there, the black and red, black and transparent. So black is going to be negative.
Lay one fiber pad flat on the black plastic.
So first you have to place your pad. We are doing in water, so it will be a buffer so that no bubbles will
Place it
stay.
on top of the pad. Be careful to avoid any bubbles between the pad
This is
and the
again
paper.
a type of
Make certain the
blotting
buffer
sheet.
covers the paper.
Wattman number 1 filter. Now next is
The
your
gel.
gel.
Carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper.
Okay, so now you have placed your gel here. So now over it you have to place your membrane.
Next, you
Use a
will apply
nitrocellulose
a
membrane.
piece of nitrocellulose membrane.
It must be appropriate to the size of your gel.
And wet the membrane with blotting buffer.
This also you need to soak in the buffer first.
Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately.
So this after the size you have to place it
Using a
in size.
roller, remove any
Now
air
with the
bubbles
roller you
between
have to
the gel
remove
and the membrane.
any bubble. So I mentioned if bubble is there, that is going to block your path. So you can't allow bubbles to stay there. Then next is again you have to place one more
Place a second sheet of wet blotting
blotting
paper
sheet
on top of the nitrocellulose membrane.
over it. And next is the pad.
Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clip it to
Now
the
close
black
it. So
plastic
tightly that
side
weight we
by
are talking about
sliding
here.
over the white clip.
We are putting it very tight, so
This tight
gel
fit
and
squeeze the sandwich together.
your membrane become very close to each other. So no air gap should remain. Now you have to place
Insert
the
the gel
cassette
holder
into
into
the
the inner
chamber.
module. Make certain that the
So
black
there
side
is
of
a
the gel
black
holder
and
is
red
next
is
to the
there. So
black
black
side of
portion
the
of
module.
your cassette should be toward the black side of
Place the
your
inner module into
chamber.
the electrophoresis chamber.
Then put into the
Add a frozen cooling unit
this
and
is for
fill the
cooling.
chamber
So
with
your
blotting
temperature
buffer
is
to the
high.
level of the white clip on the gel
And
holder.
then put the transfer buffer.
Place the lid on the
Like negative
electrophoresis
negative.
chamber.
So negative 14 will be negative to the positive.
Connect the electrical leads to the power supply.
So this is the same
Making
as
sure
the
the connections are
electrophoresis.
correct. Red to
This is
red
the
and
polyfilament
black to
derivative.
black.
This is also transfer. Also you can use the electrophoresis.
Turn on the power supply and run the plot at 20 volts. If a timer is available, set it for 2.5 hours.
The time you kept.
When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner
When
module.
the transfer is done, remove it.
Open the module and place it in a container filled with blotting buffer with the black side down.
Opening is also similarly as you have done the closing. So remove your
Starting with the
carefully
first
remove
fiber
your
pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the
So now
membrane.
how to confirm that the protein being transferred? So for that what we are using a ladder which is going to be colored. So if you see the color is going to be, you can see
Scope
the
green
bands,
stain
color bands
standards
are
have been
there on the
transferred
gel.
and can be seen on both sides
This
of
is
the
an
membrane.
indication that gel got transferred. So the color which is more intense towards side, which
You
is
can also see that there are
surface
no
to be in
longer any
touch with your
proteins on
gel.
the gel. Immerse the membrane in 25 milliliters of blotting solution.
Now you
And
have to put
incubate
this
it
into
for
the blocking
15 minutes
solution.
at room temperature
That is
on a
skim
rocking
milk.
platform.
Now the blocking is done.
Pour off the
Now we
blotting
can pour
solution.
off the blocking, so you can see white color. This is a milk solution. And now you have to wash it. Washing is necessary to remove the extra protein which was there.
Add 10 milliliters of primary antibody.
After washing, you can add the primary antibody.
Incubate for 10 to 20
Again, you
minutes
need to
on a
include the
rocking
primary
platform.
antibody.
The platform should be set at a faster setting to
Surface
ensure
got
constant
exposed
coverage
with
of the
the
membrane.
antibody. Antibody will go and bind specifically to your protein of your interest.
Pour off the primary antibody.
And discard the primary. Then washing is required, so wherever the primary antibody is there, if any non-specific thing is specific primary antibody is there, you need to remove. If you don't remove, then you may get a non-specific result also. Washing should be 2-3 times.
Add another 50 milliliters of wash buffer to the membrane and let it
So
wash for
the
3
aim of
minutes
the
on a
washing this
rocking
is to
platform
remove
at a medium speed setting.
any non-specifically associated your. So you can wash it, leave it for 10 to 15.
Discard the wash buffer. Add 10 milliliters of secondary antibody.
Now after the washing is over, then you need to add secondary antibody. Secondary antibody as mentioned is going to be
At a fast
of
speed.
your either chromogenic, radioactive, or fluorescent.
Pour off the secondary antibody.
The secondary, so here also you need to wash. Washing step is
Rinse the
necessary
membrane
also.
quickly in 50
So wash
milliliters
with the
of
wash
wash
buffer.
buffer. And discard
After the
the wash
addition of
buffer.
both antibodies, you need to go for the washing. After the blocking, you may need not to wash, or if you wish to, you can go for wash. But after the primary and secondary antibody adding, it is must to wash. Because if you don't wash, then the non-specific signal may remain on your membrane surface.
Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on a rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes.
Now the next is visualization. So visualization, you need to add substrate. So substrate will allow the reaction to occur. And wherever the proteins you can see, the chromogenic substrate is the colors. So where the bands are there, they are protein of your
Air
interest.
dry for 3 to 60 minutes and then cover in plastic wrap for storage.
So this is how you perform Western blotting and Western transfer. Okay, so now our time is also come to end. So I'm ending today's session now. Please turn on your camera for attendance. And next session on Friday, we are going to talk about discussion for your module number 2. If you have any specific question regarding northern, western, southern blot, I will be there to give you the answer to your query. And I will also share you the slides on your LMS. And also any other study material that you will be, I feel that that will be good for you to study. I will share you that also. Okay, so please turn on your camera. By I'll call your name. Adish.
Present, sir.
Atoiba,
Present, sir.
Ayushi,
Present, sir.
Demetrius,
Present, sir.
Devanshi, Chika,
Present, sir.
Manvi,
Present, sir.
Kshitij,
Present, sir.
Ashwini,
Present, sir.
Amrita,
Present, sir.
Nandini, Nehal,
Present, sir.
Rupal,
Present, sir.
Saiba.
Present, sir.
Nehal, Rupal, turn on your camera. Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir.
Yuvraj,
Present, sir.
Vivek,
Present, sir.
Yukta.
Present, sir.
Okay, so Nandini, it is a regular issue. Always you are saying that you are typing in the text box that your network is not working. Get rectify it. Next time I won't listen that your network is not working. Okay, you have the network issue. Okay, so thank you all. Stay safe, stay happy. So next session, please read that slide that I'm going to share with you, and also the study material, and come up with the questions. And after that, going to be a test for the model number 2, and then we'll move to the model number 3. Okay. So thank you all. Stay safe, stay happy. Good day.
Thank you,
Thank you, sir.
Thank you, sir.